So I'm here in Estoril attending the ESH Translational Research meeting in AML. And the topic that I'm going to talk about is FLT3 inhibitor therapeutic combinations. Now we've got a variety of targeted agents for FLT3 on the market now. And I think the challenge in the future will be to understand how best to use them. And the reasons that we might combine certain agents to address emerging clinical resistance mechanisms. So one thing that we've known for a number of years is that it's very important to have an inhibitor that can address all potential drug resistance causing mutations within FLT3 itself. But now we have some inhibitors like that, including the recently FDA approved drug gilteritinib. And as these better inhibitors or even pan-FLT3 inhibitors have come to the market, we've begun to see some off-target resistance mechanisms, such as activation of the RAS/MAP kinase pathway. There's also been a variety of other preclinical mechanisms identified that can cause resistance, such as stromal activation of cytokine signaling, upregulation of anti-apoptotics or PIM kinases, and there's actually a large number now of combination clinical trials that are currently accruing. These both include combination with high-intensity chemotherapy, low-intensity chemotherapy, and a variety of targeted agents. And I think the challenge for us will be to really understand how we can effectively utilize combinations, because after all, there's only so many drugs that we can give at any given time. Will these combinations be effective? Will they be tolerable? Are there ways that we can sequence them in a way that creatively to make them more effective and potentially better tolerated by patients? And I think I'm going to challenge our field actually to think outside the box and see if there's a way beyond just combining drugs that we can potentially think about how we can creatively use new ways of giving drugs, such as giving one agent sequenced with another agent to knock off individual clones to deal with the emerging heterogeneity of resistance mechanisms that we see. And I hope for the next ESH annual meeting that we'll have more answers than just throwing a bunch of drugs in a box and handing them to a patient.